Okay, so this is chapter 6, part 2 video. We're talking about metabolism. We talked about catabolic and anabolic reactions, endergonic and exergonic reactions. We talked about energy in general, and energy profiles for those types of reactions. And we said that in order for a reaction to proceed spontaneously, the reaction must have a negative change in free energy value, or a negative change in G. And those types of reactions are reactions that are exergonic, energy releasing. Okay, and so that's going to bring us to the laws of thermodynamics. Okay, and again, we're talking about energy here. So the first law of thermodynamics states that the total amount of energy present in the universe is a constant. It always has been, it always will be. There's not an infinite amount of energy. Okay, there's a limited amount. Energy cannot be created and it cannot be destroyed. It can only be transferred from one thing to another or transformed from one form to another. Okay? We see this, right, in living organisms. So we say that plants, right, photosynthetic green plants, producers, will harness energy from the sun, right? They'll capture it and then they'll transform that energy during the process of photosynthesis to chemical stored potential energy found in the bonds of glucose, food molecules. That's an energy transfer from the sun to the plant, and it's also an energy transformation, right, from wave energy to stored potential chemical energy found in the bonds of food. The same thing happens, right, with cellular respiration. Cellular respiration happens in plants, okay, so that wouldn't be an energy transfer. But it gets transformed for sure, right, from being stored in the bonds of glucose to ATP. Energy gets transformed. When we talk about consumers like us, right, we eat either other organisms that eat plants or we eat plants directly. And when we do that, we obtain stored chemical potential energy in the form of food, right, from those organisms. So that's an energy transfer from the plant or the producer to the consumer. And then again, we have a transformation when we talk about cellular respiration. So we've already seen how energy gets transferred and transformed, and we're going to see more of that, right, when we get into those processes a little bit deeper. Why do organisms need this constant supply of energy? In order to maintain homeostasis. So in order to live, organisms will need a constant supply of energy from their surroundings. Okay, and again, photosynthetic organisms or producers are going to get that energy from the sun. Organisms like us, consumers or decomposers, are going to get that energy from other organisms. And we need that in order to maintain life functions. So we need a constant supply of energy. Okay, so here we go. Let's start actually on the right here. This figure's kind of funny. So here's our sun. Right, the sun's beating down on our planet every day, and some of that energy from the sun will be captured by photosynthetic organisms and will be transformed. So that's the transfer, right, from the sun to the photosynthetic organism, and then it gets transformed. It changes, right, from light energy, or energy in the form of waves, to chemical energy, energy stored in the bonds of glucose. And that chemical energy represents stored potential energy. Now, those plants can use that energy themselves, right? They could break down that food for ATP, or organisms like us, these little tiny humans, will eat, in this case, we've got a bad example, ice cream loaded with sprinkles, or this could be, you know, an orange or an apple or a piece of, other piece of fruit or a vegetable or meat. Okay, and we're going to obtain chemical energy from those food sources, and yes, even from ice cream, we can obtain chemical energy, and we will transform that energy into ATP, and then that ATP can be used to do work, and that would be a transformation into from chemical energy into kinetic energy, the energy of motion, the energy being used to do work. In this case, to ride a bike, right, to move those muscles. Okay, so energy cannot be created, nor can it be destroyed. It can only be transferred and transformed. The energy of the universe is a constant. Energy is being transferred and transformed every day. Eventually, the energy supply to our planet will run out. Okay. Now we know that energy is not does not get recycled in an ecosystem, and so therefore that's when the second law comes into play. No energy transfer is 100% efficient. Okay, so when our cells in our body do cellular respiration, they're not going to harness or be able to transform every bit of energy held in the bonds of glucose. In fact, a large amount of that energy will be lost. Okay, so no energy transfer or transformation is 100%. Instead, some of that energy gets lost as unusable energy. Okay, energy that dissipates, it's let off usually in the form of heat, and we say that that energy is going to increase the entropy. And when I think of entropy, I think of disorder of the universe or unusable energy. You can think of living organisms as islands of low entropy organization, organized living things, in this kind of sea of rising entropy. So every time our cells use energy, they're using energy to maintain order, okay, to maintain homeostasis, but our surroundings are constantly going to be increasing in entropy. Okay? So what do you need to know here? You need to know that the first law states that energy is not created nor is it destroyed, instead it is transferred and transformed. Okay? Living organisms need a constant supply of energy in order to maintain homeostasis. The second law states that no energy transfer or transformation is 100% efficient, and that some of that energy will be lost as heat. Okay? As energy is used, heat or entropy will be on the rise, okay? And that entropy represents that unusable energy. So the amount of available, usable energy on the planet is constantly decreasing, and the amount of entropy in the universe, not just on the planet, is on the constant rise. Okay. So we've got our first two laws there. Let's talk about ATP. Now, we alluded to this a little bit in that first video. We talked a little bit about ATP. We said that ATP is the energy currency or the energy source that our cells, the cells of living organisms, are able to use. We cannot use glucose directly. Plants cannot use energy from the sun directly. It first has to be transformed. And ATP is going to be the molecule of choice. This is the molecule that can drive processes inside of our cells that require energy. ATP is a small, simple molecule. It's going to store energy short term, okay? So we consider it to be potential energy stored in the bonds of this molecule. It's going to allow for quick bursts of energy, again, that are going to drive cellular work. It is the primary energy source for cells. It's the kind of energy that your cells can use directly. And again, it's going to power most processes inside of our cells that require energy. This is what the molecule looks like. I don't need you to memorize the structure. I just want to show it to you. So here is adenosine. Okay. It's a nitrogenous base attached to a ribose sugar. Okay, and then here's the important part of the molecule. We've got 3 phosphate groups. 1, 2, let me actually grab my pen here. Okay, so this part of the molecule is called adenosine. Okay. And then we have 3 phosphate groups. 1, 2, it's hard to draw on here, let me use my finger. 3, okay. Some books will refer to them as phosphate group 1, 2, and 3. This third phosphate group is always called the terminal phosphate group or the end phosphate group. Okay, and then some books will refer to them as the alpha, beta, and gamma phosphate group. Either way is fine. Now that terminal phosphate group can come off and then can be rejoined. Okay, the energy between phosphate groups 2 and 3, that bond there connecting them, that's going to be the energy that is going to drive cellular work. Okay, that's the stored chemical potential energy there in that last terminal phosphate group. Okay, here it is in words. Again, ATP is composed of adenosine, which is made up of an adenine and a ribose. And then the 3 phosphate groups. Again, we can call them alpha, beta, and gamma, or 1, 2, and 3. It's the bonds between phosphate groups, really 2 and 3, that are really, really important. Okay? Okay. So here's just another picture of the molecule. So again, we've got we've got adenosine, which is an adenine and a ribose, and we've got our 3 phosphate groups, 1, 2, and 3. Okay, here it is simplified. I like this better. We don't have to see all those bonds. Water, through the process of hydrolysis, okay, will split the molecule right there, and the products of that reaction will be adenosine diphosphate, or ADP for short, inorganic phosphate, PI for short, and the most important thing is the energy held in that bond is now released. Okay, so the breakdown or the splitting off of that terminal phosphate of ATP releases energy. Okay, if we were to do the reverse of this, so remove water and put this phosphate back on to get this molecule, that would require energy. Okay, that's actually what happens during cellular respiration. Glucose is broken down, and through a series of steps that we're going to learn all about, inorganic phosphate, PI, gets added back to ADP. Okay, and now we've created that short-term energy molecule called ATP. Okay? Okay, so how is this used? Let me give you an example. This is called energy coupling. Okay, so if we break the terminal phosphate off of ATP, the energy that's released, we know, is about 7.3 kilocalories per mole, okay, or 32 kilojoules per mole. So the reaction, the breaking off of that terminal phosphate from ATP is a very exergonic reaction. It's an energy releasing reaction. Okay, now that energy that's released, it can be used to drive endergonic reactions, anabolic reactions, buildup reactions that require energy. And that's called energy coupling. Again, we're going to be using the energy from ATP to drive other reactions. Okay, so that's shown here. So if you look at this second reaction, if we take glucose and fructose, okay, those are 2 monosaccharides, and we put them together, okay, that process will be called dehydration synthesis. Remember, we learned about that back in chapter 3. We will make the disaccharide sucrose. Okay, we will also release water in that process. That reaction, going from 2 monosaccharides to a disaccharide, that is an endergonic reaction. It's a buildup or anabolic reaction. We're building something larger from 2 smaller subunits. If we look at the energy profile for that reaction, the change in free energy is a positive 6.5 kilocalories per mole. So that is not a spontaneous reaction. It's going to require energy. Where is that energy going to come from? It's going to come from ATP. The energy that gets released when ATP is split will be used to drive this reaction. Okay, that's shown here. So this final reaction shows energy coupling. Glucose plus fructose plus ATP will yield sucrose, our disaccharide, plus adenosine diphosphate and that inorganic phosphate. That's ATP that's been split. Overall, Okay, if we combine the change in free energy values, so negative 7.3 kilocalories per mole for the hydrolysis of ATP and a positive 6.5 kilocalories per mole for the production of sucrose, if we combine those, we get a change in free energy value of negative 1.2 kilocalories per mole. That's spontaneous, right? If the change in free energy value is negative, then it is a spontaneous reaction. So that can happen. Okay, we don't need any more energy. The energy provided from splitting one molecule of ATP was enough to drive that reaction. So again, that's energy coupling. It's taking the energy released from an exergonic reaction and using that energy to drive an endergonic reaction. Okay, and just so you know, how does this happen? It's pretty interesting. So what happens is really ATP gets split. Okay, that phosphate group, the terminal phosphate group, the third one, actually comes off of ATP and gets added to glucose. So this is actually the first part of the reaction. Glucose plus ATP yields phosphorylated glucose, glucose with a phosphate on it, plus ADP. And then what happens is that phosphorylated glucose, glucose with a phosphate on it, the phosphate then will come off and fructose gets added. Okay, so again, oftentimes we'll write, let me just erase this. We'll write formulas like this, real simplified, but in actuality there's multiple steps, right? It's a pathway. So really it's this. Okay, but we write it like this to show a summary of that reaction. But really there's steps. First, a phosphate comes off of ATP and gets added to glucose. And then the phosphate comes off and fructose gets added. And we end up with sucrose. Okay, so again, that's called energy coupling, using the energy released from one reaction to drive an uphill reaction. What else does energy get used for? I talked about some of this, I think, in the last video, but I just wanted to show you, and we've already talked about this back in chapter 5. Remember the sodium-potassium pump? That was our example of an active transport. In this case, what happens? We've got our cell membrane here, we've got the outside of the cell or the extracellular fluid, and then we have the cytoplasm on the inside, and we need to build up an electrochemical gradient. And so what's going to happen is this blue thing in the membrane, that's the protein, right? It's a carrier protein. It's going to pump 3 sodium ions out and pump in 2 potassium ions. And it's pumping those ions against an electrochemical gradient, okay, to build up that gradient. And so how does it do that? It needs energy. Where does that energy come from? Well, that's where ATP comes into play here as well. So what will happen is ATP, that terminal phosphate, right, phosphate number 3, will come off of ATP and will be added to the carrier protein. And so now we're left with ADP and inorganic phosphate, PI. And the transfer of that phosphate is equivalent to the transfer of energy, right? So that negative 7.3 kilocalories per mole of energy gets transferred to the carrier protein. The carrier protein changes shape, okay, and then that terminal phosphate group comes off of the carrier protein. Okay, and the process is done. So that energy is going to be used to change the shape of that protein carrier. Okay. I just wanted to show you this picture. I like this. This shows ATP. It's kind of like a rechargeable battery. Okay, that's kind of how I think of this molecule. It stores energy, it releases energy, it needs to restore its energy. So here we go. We've got exergonic reactions. Okay, energy releasing reactions like cellular respiration. They're going to be used to take ADP and add to it the terminal phosphate, inorganic phosphate, to make ATP, adenosine triphosphate, right? Adenosine with 3 phosphates added to it. That ATP will then be used to drive endergonic reactions, or anabolic reactions, those reactions that require energy. Okay, reactions or any processes that require energy. Okay, in that process, the terminal phosphate gets donated and gets split off of ATP. And so what you're left with at the end of the process that requires energy, again, is ADP, adenosine diphosphate, and that third phosphate group that's now not attached, so we call it inorganic phosphate. And again, these will be put back together through exergonic reactions, and the cycle continues. So it's a cyclical event here. Okay? So again, ATP, it's a small molecule made up of adenosine with 3 phosphate groups. That terminal phosphate will come off to donate energy. It will be put on back on through energy releasing processes. This is the energy currency, the type of energy that your cells can use, that all living things, their cells can use. We cannot use energy from the sun directly. We cannot use energy stored in the bonds of food like glucose directly. Instead, that energy has to be transformed into ATP before it can be used to drive cellular work. So ATP is the middleman. Okay, so take a break here. You're going to go ahead and you're going to answer these questions. So again, talk about those first and second laws of thermodynamics. You are going to talk about ATP, talk about its structure, how it works, and what energy coupling is. When you're finished, you are going to come back, and I've got some questions for you. Okay, so first question. The first law of thermodynamics states that, pink, no energy transformation is 100% efficient. Blue, energy cannot be created, nor can it be destroyed. Green, energy in the universe is increasing. Or yellow, entropy of the universe is increasing. So which of these describes the first law of thermodynamics? And again, you're going to try not to look back. Okay, so hopefully you answered blue, right? So the first law states that energy cannot be created, nor can it be destroyed. It can only be transferred and transformed. In pink there and in yellow, those describe the second law, okay? All right, next question. In energy coupling, ATP is used to drive active transport? Is that coupling? Is it used to drive bulk transport? Or green, is it used to drive endergonic reactions? Or yellow, is it used to drive exergonic reactions? So which one of these describes energy coupling? So hopefully you answered green. ATP is going to be released by exergonic reactions, and it's going to be used to drive endergonic reactions, build up anabolic reactions that require energy, right? Those uphill reactions. Okay, next question. Which of the following is not true of ATP, adenosine triphosphate? Pink, it transfers its energy by transferring its terminal phosphate. Is it true or not true? We're looking for the not true statement. Blue, it is produced by exergonic reactions. Green, it is a long-term energy storage molecule. Or yellow, it is the primary energy currency for cellular work. Which one is not true? Okay, so the one that is not true is green. ATP is a short-term energy storage molecule. Cells will not store energy long-term in this form. Long-term energy storage, happens in molecules like, fats, triglycerides, or in molecules like starch if it's a plant, or glycogen if we're talking about animals, right? Storage and carbohydrates or lipid molecules. ATP is a short-term energy storage molecule for short bursts of energy. Okay, that brings us to our next section where we're going to talk about enzymes. And we've already actually introduced ourselves to enzymes in the first video. We talked about both exergonic and endergonic reaction profiles. And we said that no matter what, all reactions have activation energy, right? That energy barrier to get reactants to the transition state. And we said that the role of enzymes in cells is to decrease that activation energy, to lower the energy barrier so that the reaction can proceed. Okay, so here we go, enzymes. So cells will regulate the speed or the rate of chemical reactions with these molecules called enzymes. Enzymes can increase the rate or the speed of a chemical reaction without being used or consumed by the reaction. Okay, they can be recycled by your cells. Your cells can use enzymes over and over and over again. And again, even spontaneous reactions, that term spontaneous does not mean fast. And so enzymes will speed up the rate of all reactions, endergonic or exergonic, spontaneous or not spontaneous. So even if a reaction has a negative change in free energy value, enzymes are still going to increase the speed. They're still going to be used to decrease that activation energy. Here's an example of an enzyme. This is an enzyme that we're going to use in the lab. The enzyme is catalase. Notice the ending there, ASE. Most enzymes are going to end in ASE. That's indicative of an enzyme. And also, most enzymes are proteins. The next sentence down describes that as well. We do have some type of RNA molecules that have catalytic activity, but most enzymes are going to be proteins, and most will end in ASE. So catalase is an enzyme. Another name for an enzyme, if we're talking about biological systems, is catalyst. Okay? We get a little bit confused, especially when we do this lab, because catalase is the catalyst. The terminology is a little bit confusing here. So catalase is an enzyme. It's found in our cells. It's found in the cells of the organism that we're going to use in the lab, yeast, or in our case, if we're doing this lab at home, the organism that we're going to actually use is the potato. Okay, so it's found in all organisms. And catalase has the highest known catalytic rate. Very, very nice to study, very easy to watch. The job of catalase is really protection of cells. What catalase does is it will take hydrogen peroxide, which is H2O2, okay, that stuff in your medicine cabinet in the brown bottle. That's also produced inside your cells as a byproduct of many reactions. If catalase was not present, hydrogen peroxide, the breakdown of hydrogen peroxide is a spontaneous reaction. Hydrogen peroxide will break down into water and oxygen, but it's not a very fast reaction. And so when hydrogen peroxide is produced inside of your cells, it can be toxic. It can damage your cells. And so your cells don't have time to wait for hydrogen peroxide to break down into water and oxygen. Instead, your cells need to speed up that breakdown. And so catalase is going to be the enzyme that is going to speed up that reaction. It will actually bind to the hydrogen peroxide. It will create tension and contortion of the bonds of hydrogen peroxide, causing it to reach the transition state very readily, right? It has a very high catalytic rate. And then those bonds will be broken and water and oxygen will be then released from the catalase enzyme. Okay, so catalase is the enzyme that we're going to be studying in the lab. It has the highest known catalytic rate. Its job is to break down this toxic molecule called hydrogen peroxide, H2O2. Okay. Next slide, I wanted to show you again, even strongly exergonic reaction, even spontaneous reactions, can be slowed down or prevented from occurring because of the activation energy. Let me grab my pen here. Okay, so here's our energy profile. If we look in red, this is the energy profile for this exergonic reaction without the presence of an enzyme. So I'm going to just kind of highlight. Oop, no, I'm not. Here we go. I'm going to hide. Nope, I'm not. What happened to my pen here? Sorry, lost it. Okay, so in red, again, I'm just going to kind of trace over it. That's the reaction profile for this reaction without an enzyme. So if we look at this line right here, this is the activation energy, the E of A, without the enzyme present. Okay, notice here, the free energy change, the delta G, is negative, right? That means that the products have less free energy than the reactants. So energy is released through this reaction, but there is still that activation energy, that hump, that barrier in the very beginning. Okay? In gray or black, trace over it really quick, that is this reaction with the presence of an enzyme. So notice that the change in free energy value doesn't change. The same amount of energy is released. The energy of the reactants versus the energy of the products is the same. The only difference here is that the energy of activation, that barrier, right, that is there, has decreased. Okay, at the very top of this E of A, which is not written here, I think it might be on the next slide, that represents when the reactant molecules reach the transition state. Okay, after they reach the transition state, they will form products very readily. Okay, so it's really the energy required to break the existing bonds. Okay, that's going to start the reaction. Oh, it's not here. Okay, I thought it was. Here is the reaction profile for an endergonic reaction, just to show you. Again, the red dotted line in this case is with a catalyst or with an enzyme, and in purple it's without the catalyst or without the enzyme. You can see this is an uphill reaction. So the change in G, which is represented, I think by this red arrow. This one's a little confusing. That's the change in free energy value. Okay, so there's an increase in free energy, so that's a positive delta G. So this is a non-spontaneous endergonic reaction. Okay, and then you can see here, that's the activation energy, with and without an enzyme. So with the enzyme, that red dotted line, you can see that there's a drastic reduction in the activation energy. Okay, that's, again, the energy required to reach the transition state, to break the bonds found in the reactant molecules. So whether we're talking about an exergonic or an endergonic reaction, we're going to see this requirement for activation energy. Okay, enzymes will lower that. Okay, how do they really work? Well, what happens is the enzyme actually will bind to the reactants. Okay, so in the case of hydrogen peroxide, the enzyme, catalase, binds to the hydrogen peroxide. So again, the enzyme is catalase. The hydrogen peroxide we call the substrate. So there's that term right here. Okay, and that's going to form something called the enzyme substrate complex, or the ES complex. Okay, once that happens, once there is that binding, we will reach the unstable intermediate, the transition state, and then products will be formed. Once the products are formed, they get released from the enzyme substrate complex. So here's a little set of reactions, just to show you that summary. So again, you have your enzyme and your substrate. We'll talk about catalase. So we have catalase is our enzyme. The substrate is hydrogen peroxide. They are going to bind, very specifically, to form the enzyme substrate complex. Those bonds become weak. Water and oxygen are formed. Okay, and then they get released from the enzyme substrate complex. And so here we have our enzyme. Catalase is going to be available for the next round of reaction, and the products get released. So that's that water and oxygen. Okay, so enzymes work by physically binding to their substrate. So here we go. In blue, we have our enzyme or our catalyst. Okay, in our case for lab, that would be catalase. And then we have this reactant molecule, which we're going to call the substrate. Again, in lab, that's going to be hydrogen peroxide. And then inside of here, there is a very specifically formed region called the active site, or the substrate binding site. That's where the substrate will bind. This is really important. If you remember back to chapter 3 when we talked about form equals function, especially with proteins. Remember, most enzymes are proteins. When a protein folds up into its proper three-dimensional form or shape or configuration, that's when it's going to work. If it doesn't fold properly, it won't work. So this enzyme has to fold perfectly so that that active site is a very specific shape, the shape for its substrate. You may have heard of lock and key. That's being shown here by this figure beautifully. Okay, you've got your enzyme and you've got your substrate, and the substrate has to fit perfectly. Okay, if it's not the right shape, it can't get in there. So if the protein doesn't fold properly, this doesn't work. Okay, it's all about form equals function. So when that active site is open properly, you get your substrate, it binds, and then what's going to happen is you have your enzyme substrate complex, and the enzyme will actually kind of hug or fit perfectly around that substrate. That's called induced fit. And you can see that here. You can see how there's been a change in shape of the enzyme after that substrate has bound. Okay, again, that's going to be called the enzyme substrate complex. And here's that term induced fit right here. Oh, hold on a minute. Okay, I don't know what happened. So here's that term induced fit. Oh, again it happened. Okay, my computer's being wonky, so I'll just use the mouse. So again, enzymes will bind to substrates via that region on the enzyme called the active site. They're going to position the substrates close together, or they'll take, if it's just one substrate, they'll take and they'll kind of bend the bonds between the atoms. And that's going to speed up the reaction. So that physical binding there is going to speed up the reaction. Again, there's that term induced fit. The binding of the substrate to the enzyme causes a bit of a change in shape to the enzyme. And again, that's going to be required to distort the bonds, the chemical bonds of the reactant molecules, the substrate. You can kind of think of it as the enzyme kind of giving the substrate a little bit of a hug. Okay, it brings it right in. Now, the enzyme might speed up the reaction again by just kind of contorting those bonds. It may also, if there's multiple reactant molecules, it may bring them just closer together, so closer proximity, or it may just orient them perfectly. Okay, all of that will lead to speeding up the chemical reaction, an increase in rate. Okay, so here's just a little picture showing the steps of this process. In this case, our substrates here are the green and red little molecules. Okay, so we have two reactant molecules, sometimes just one, sometimes two or more. Okay, the enzyme is shown here in purple. We have the substrates that bind to the enzyme. We have the induced fit that happens. And again, that's going to be called the enzyme substrate complex. There will be some contortion of bonds. The substrates might be aligned properly in good proximity to each other. And that's going to cause products to form. Products will then be released from the enzyme. And the enzyme will be now available to the next set of substrate molecules. Okay, so again, enzymes are not changed or consumed in the process of the reaction. Instead, they're really recycled and available for the next round. Okay. I feel like I wanted to say one other thing on that previous slide. So they're not changed or consumed. They're recycled. They can be used multiple times. Oh, it'll come to me. Okay. Something else that I wanted to add. Enzymes are going to work best, or they'll be at their optimum performance in very specific environmental conditions. And if you get out of the range for an enzyme, then the enzyme's activity or productivity will decrease. Okay? So let's look at these examples here. In the first graph that we have, and again, I think my pen's being wonky. So in the first graph here, we're talking about temperature. We've got two different enzymes. One of the enzyme is a typical human enzyme that's going to operate best at human body temperature, which is roughly 37 degrees Celsius. Okay? The other enzyme that we have comes from a thermophilic or heat-loving bacteria, which operates best at 77 degrees Celsius. That's pretty hot. Okay? And we're looking at the activity now of these enzymes. So we're looking at whatever reaction they catalyze, and we're looking at the rate of the reaction. So in black, we have our human enzyme, and in red, we have our thermophilic bacteria enzyme. And so you'll notice that the activity of the human enzyme is very low, right? Nothing, at 0. As we increase temperature, it slowly starts to creep up in activity. When we reach 37 degrees Celsius, that's when this enzyme is going to work best. It peaks. If we increase temperature, notice how the activity of that enzyme drops off real fast. Okay? Let's look at the heat tolerant. The heat tolerant enzyme is not going to start working anywhere. Between 0 and 40, it's still not working. But as we increase temperature above 40 degrees Celsius, it starts to creep up. It starts to have some activity, but its optimum activity is at 77 degrees Celsius. And again, if we go above that, look how fast it drops off. Okay, so the optimum rate or the optimum temperature for a particular enzyme is going to depend on the organism that the enzyme comes from. Okay, if it's a human enzyme, it's likely going to work at human body temperature, about 37 degrees Celsius. But if it's coming from an extremophile, like a bacteria that lives in extreme heat, it's going to operate a little bit higher. The enzymes that we're going to study in lab are from organisms that do not live in any extreme temperature. Okay, so they're going to work best around the 37 degrees Celsius range as well. Okay. Let's look at, before I go on, I want to talk about, remember how we talked about form equals function? So we said that enzymes have to have a proper three-dimensional shape in order to bind to substrate molecules. So why does temperature affect that? Well, in order to fold properly, the enzyme has to be at the right temperature. If you take the temperature outside of its optimum or outside of its range, what actually happens is that the enzyme just unfolds. Okay, so in this case, for the human enzyme, as we go above 37 degrees Celsius, it's too hot. The protein just unfolds. We call that unfolding. We say that it becomes denatured. Okay, so that term denature, which is down here for you, that means to unfold. And if a protein is not folded properly, we know that it can't function. The active site will not be the appropriate shape or size, and so therefore the protein can't function. We can say the same thing for pH. So enzymes will have an optimum pH. We talked about pH already. An optimum pH where they're going to work best at. For an enzyme, a human enzyme found in the stomach, this enzyme is called pepsin. The stomach is an acidic environment, and so this enzyme prefers a pH of about 2, even a little bit less. Okay, that's where it operates best at. If we get outside of that range, enzyme activity goes down. Why? The enzyme unfolds. Doesn't work right. If we look in the intestine is more of an alkaline environment. This particular enzyme is called trypsin, and it works best at a pH of about 8. That's where it peaks. Again, if we get outside of that range, or if we were to take trypsin and put it into the stomach, it wouldn't work. It would unfold. Okay, so every enzyme is going to have optimum environmental conditions where it will work best at. And that might be temperature, it might be pH, okay? And again, if we get outside of the range for that enzyme, the enzyme doesn't work. And sometimes that's due to the enzyme unfolding, denaturing. Okay. Just a little bit left here. Let's talk about enzyme regulation. So we know that we can affect enzyme activity or rate by changing environmental conditions, but there's also other ways that we can regulate enzymes or regulate enzyme rate or activity. We have molecules called competitive inhibitors and molecules called non-competitive inhibitors. And both of them, as the name implies, inhibit or prevent enzyme activity, or decrease at least enzyme activity. A competitive inhibitor would be a molecule that binds to the active site of the enzyme, blocking substrate binding. So we say that this type of a regulator competes with the substrate molecule for the active site, and that's why it's called a competitive inhibitor. If it's bound, then the substrate cannot bind. On the other hand, a non-competitive inhibitor, it still inhibits enzyme activity, but instead of binding to the active site, it's going to bind to some other part of the enzyme, causing the enzyme to change shape and therefore making the active site not shaped correctly, and therefore the substrate cannot bind. Okay, we have some kind of naturally occurring inhibitors that work either as competitive or non-competitive inhibitors. There are toxins that work this way, poisons, pesticides, okay, and also antibiotics. So you've all taken antibiotics before. Antibiotics are specific to bacterial proteins, and that's why they don't affect your cells and your proteins. But what an antibiotic does is it will inhibit bacterial enzyme activity, either in a competitive or a non-competitive fashion. And if the enzymes of the bacteria don't work, the bacteria can't live. Okay, reactions cannot proceed, and therefore the bacteria will die. So those are some examples of, oh, here we go. Here's pictures. I forgot about this. So this is just showing normal binding versus competitive versus non-competitive. So in our first example here, this is normal binding. So we have our enzyme in purple. You can see the shape of its active site. The substrate is in teal. The substrate will bind perfectly to form the enzyme substrate complex. Everything goes perfect in that first example. B, this is competitive inhibition. And so the blue molecule is the competitive inhibitor. It will bind to the active site, blocking the substrate from being able to bind. Okay? A non-competitive inhibitor will bind to a site other than the active site, causing the enzyme to change shape, and therefore the substrate cannot bind. Okay, so again, that's inhibition. Okay. So again, those types of inhibitors, you know, we got poisons, we've got toxins, pesticides, antibiotics. Those all sound like really bad things, right? But your cells also kind of naturally will activate or inhibit enzymes. You know, so these are natural things that occur inside of our cells that are supposed to be there to regulate enzyme activity. And that usually is going to occur in an allosteric fashion. So we'll talk about what an allosteric fashion means. So enzymes will have a site, not the active site, but another site on them called the allosteric site. And that's where either an allosteric inhibitor or an allosteric activator can bind, changing the shape of the enzyme. An allosteric inhibitor is going to change the shape of an enzyme, causing an inactivation, where an allosteric activator causes the change of shape of the enzyme to activate it, okay, or to bring it to full activation. So let's take a look at that. Here's a picture. So in black here, we have our enzyme. Okay, and on the left, we have allosteric inhibition. And on the right, we have allosteric activation. Let's do inhibition first. So again, here's our enzyme. It's in black. The active site is shown here. It's open. This is an active enzyme. Here is the allosteric site. Okay, this is where this little orange inhibitor can bind to. And when that binding happens, it changes the shape of the enzyme shown here. And the active site is no longer the proper conformation or proper shape. And therefore, the substrate cannot bind. And so that enzyme right now is inactive. Allosteric activation is kind of the opposite. Here we have an enzyme. It's in a shape where the active site is not open. The activator will bind to an allosteric site on the enzyme. So this is a site other than the active site. And that binding, shown here down at the bottom, causes that active site now to have the proper shape for the substrate to bind. So your cells will control your enzymes in this fashion. When you want a reaction to proceed, the activator causes activation of that enzyme. When you want to shut down a reaction from proceeding, the inhibitor will bind and shut down that enzyme. Okay. Your cells only want to run reactions when necessary. Okay, so they will also control enzymes. Okay, other ways in which enzymes are regulated. Many enzymes require cofactors or coenzymes. So some cofactors are things like inorganic ions, like iron or magnesium. Those have to be bound sometimes to enzymes in order for them to work. And if they're not bound, the enzyme doesn't work. Coenzymes, these are a lot of dietary vitamins that you get every day from the food that you eat or from if you take a multivitamin. Sometimes those are required also. Okay. Enzymes are also controlled or regulated based on where they're found in your cell. So we say that enzymes are compartmentalized into organelles. Some enzymes are only found in the mitochondria, and so they're only going to do their job inside of there. They don't do their job out in the cytoplasm. Okay, so compartmentalization also controls enzymes. I just wanted to show you this one last thing. This is called feedback inhibition. Again, I said that your cells only want enzymes to be active when you want reactions to take place. Your cells shouldn't, waste energy or time or molecules on reactions when there isn't a need for the product. Okay, so feedback inhibition is a way to control whether or not a reaction proceeds as well. So let's take a look here. We've got a metabolic pathway. We've got the substrate molecule here in green, the green triangle. And we've got our first enzyme here, looks like Pac-Man, shown in black. And these will bind, and here's our first enzyme substrate complex. Enzyme 1 will then convert the substrate here, the green triangle, to this purple molecule. So this is the product of that first reaction. That purple molecule then becomes a substrate, and it will bind to enzyme 2, and it will be converted to substrate B. This is our second intermediate molecule. And then substrate B will then bind to enzyme 3, and enzyme 3 will convert substrate B into our end product here, which is shown in yellow. Okay, so that pathway will happen if there's not a lot of end product, and as long as all of these enzymes are in their active form. But what happens when the end product starts to build up? So we get a lot of those yellow squares. Do we need this reaction to proceed? Is it smart for the cell to keep this reaction going if the end product isn't being used? No, right? We want to work on a supply and demand fashion. So this end product, if it starts to build up, it will actually bind to enzyme 1 at an allosteric site, a site other than the active site in this case. And that will cause the enzyme to change shape and no longer be able to bind to its substrate molecule. And this reaction will be shut down. Okay, now if the end product starts to get used, well, then the enzyme will no longer be blocked and we will turn this on. Okay, so this is called feedback inhibition. It's when the end product shuts down a reaction very early in the pathway usually at the very, very beginning, causing that pathway to stop or to be halted. Okay? I remembered what I wanted to tell you earlier. I want to also kind of hit home that enzymes are very specific to their substrates. So in this case, enzyme 1 will only bind to and act on the green substrate. It cannot bind to anything else. Its active site is not properly formed. It's not going to catalyze any other reactions. The same thing when we talk about catalase, right? Catalase will bind to hydrogen peroxide and speed up the breakdown of that molecule, but it's not going to bind to any other substrate molecules. It's very specific. Every enzyme is very specific to the reaction that they catalyze. Okay, your turn. So here we go. Here's your list. So go ahead and hit pause, and you're going to go through and answer these questions. Again, try to not look back, right? You've read your chapter, you've done your vocab list, now you've listened to me talk about this. Try to hit pause and try to see if you really know these, okay? Of course, go back and check your answers before you submit it, but let's see if you know it. Well, when you're finished, come back. I've got a few questions for you. Okay, so first question. Which of the following is not true about enzymes? Pink, do enzymes increase the change in free energy of a reaction? Blue, are enzymes usually made of amino acids? Green, do enzymes lower the activation energy of chemical reactions? Or yellow, is each enzyme specific to the particular substrate or substrates to which it binds? So which one of these is not true? So hopefully you answered pink. They do not increase the change in free energy of reactions. Instead, they lower that activation energy. Okay? The rest of those are true. Next question. An allosteric inhibitor, so an allosteric inhibitor, does which of the following? Pink. It binds to an enzyme away from the active site and changes the conformation or shape of the active site, increasing its affinity for substrate binding. So is that true? Or blue, does an allosteric inhibitor bind to the active site and block it from binding substrate? Or green, does an allosteric inhibitor bind to the enzyme away from the active site and change the conformation or shape of the active site, decreasing its affinity for the substrate? Affinity means attraction. Okay, so is that true for allosteric inhibitors? Or yellow, binds directly to the active site and mimics the substrate. So which one of those is true? So again, hopefully you answered green. Okay, so an allosteric inhibitor is going to bind to the enzyme, not at the active site. It's going to cause a change in shape, and it's going to decrease substrate binding. Okay, next question. Which of the following analogies best describes the induced fit model of enzyme substrate binding? Okay, so induced fit. A hug between two people, a key fitting into a lock, a square peg fitting through a square hole, and a round peg fitting through a round hole of a children's toy. Or yellow, the fitting together of two jigsaw puzzle pieces. Okay, this is tricky because a lot of these describe enzyme substrate binding, right? Lock and key, the peg analogy, two jigsaw puzzle pieces fitting together. But in the question, it says which describes the induced fit. Remember, the induced fit is not just the binding, that's the initial part, but the induced fit is the actual wrapping around of that enzyme. So a hug between two people would be the best answer here. Okay, that's the last one. So this is it for this chapter. So again, you want to make sure that you have all of your Are You With Me's completed. Remember, if I ask you to sketch or draw something, make sure that you actually do that. Okay. And then you're going to scan your documents and then upload to Blackboard, hopefully in a PDF, and you can combine all of those images together. If you haven't learned how to do that yet, try to figure that out into just one document so you're only uploading one PDF. Okay, and again, if you ever need my help, come to office hours. Okay, and then when you're finished with that, you can, well, let me back up here so it's light. Okay, so when you're finished with that, you're going to go ahead and you're going to study, and you're going to go ahead and you're going to take the Chapter 6 quiz.